Can you see my screen? Can you see my screen?
Yes, we do. I think, yeah, everyone can see it.
Let's see here. Let me try. It looks like it might be cut off. Is it cut off on the right at all?
It's yeah, to me looks good. I mean, I don't know. It looks good. It's like maybe the aspect ratio is a bit strange in the sense that I can see everything here.
Let me, just to be safe, let me reshare. Okay. I see what's happening. All right, give me 1 second.
Sure.
All right, there we go. Okay, how do we look now?
Yeah, I think the sharing didn't work.
Okay, let me reshare. All right, here we go.
Now it works.
Okay, how about now?
Looks good to me.
Okay, all right, fantastic. All right, thanks so much for being patient with me while we figure out the various dynamics of giving lectures over Zoom. It's funny, it's been a year and a half and still there are bugs like this every now and then. So anyways, hi everyone, I'm Majumder, and as I was introduced, I'm a faculty member in the Computational Health Informatics program at Boston Children's Hospital and Harvard Medical School. Today, I will be discussing a few of my research group's studies at the intersection of machine learning and public health. I've been applying machine learning methods to public health problems for about half a decade now and have responded to several global infectious disease crises since 2014, including Zika in Central and South America and Ebola in West Africa. But my ongoing research regarding the COVID-19 pandemic will, of course, be front and center in my talk today. We'll kick things off with a bit of context regarding the ongoing pandemic. Then from there, we'll move on to 3 of my research group studies from the past year that employ machine learning methods to better understand COVID-19. Because we have limited time today, I'll keep my overview of each study high level, covering the research questions first, then the methodology used to address them, followed by findings, policy recommendations, and related work. For those who want to learn more, all 3 studies have been published already and are open access, and have included citations throughout my slide deck to make it as easy as possible to find them. I plan to speak for about 45 minutes to an hour. We'll see how that goes, followed by an extended Q&A session at the end of the lecture to accommodate the class and the class's needs. That said, feel free to shoot me questions on Twitter as well if you prefer. My handle is at the top left of each slide. Now let's get started. As many of us on the line are aware, there have been nearly 200 million cases of COVID-19 reported worldwide since late 2019, caused by the novel coronavirus SARS-CoV-2, shown on the right. These cases comprise only a fraction of total infections, though, due to something we like to call underdocumentation. Underdocumentation has many causes, including the fact that many SARS-CoV-2 infections are mild or asymptomatic in nature, as well as insufficient volume of lab testing in the early days of the pandemic, a problem that persists in under-resourced communities, both in the United States and around the world today. Another reason for underdocumentation in the early days of the pandemic was simply because cases were often misclassified. The earliest cases of COVID-19 presented as pneumonia, which can have many causes beyond SARS-CoV-2, and may thus have been diagnosed as something else at the time. As is an issue with any novel pathogen, there's a learning curve associated with discovering even the most basic attributes of its characteristics in humans and its behavior in populations. For instance, though transmission of SARS-CoV-2 started in Hubei, China in late November 2019, as far as we know, it wasn't until late January 2020 that sustained human-to-human transmission was suspected, which is when my group began researching the novel coronavirus and its epidemiological characteristics. Our early work involved assessing the transmissibility potential, often described as the basic reproduction number, or R naught, R sub 0 for short, associated with SARS-CoV-2 in Hubei, China. The basic reproduction number is often defined as the average number of secondary infections caused by a single index case in a fully susceptible population. As a result, it is population specific and varies across contexts, which will be very important to remember as we proceed through this lecture. One can imagine, for example, populations where individuals have many daily contacts may exhibit higher values of R naught than populations where individuals have fewer daily contacts. In a population where R naught for a given pathogen is estimated to be greater than 1, we expect that the pathogen has epidemic or pandemic potential. However, because R naught cannot be directly observed, we need to model it. There are a lot of different models that let us estimate R naught, but early on in this pandemic, we had so little data available that we decided to take a simple phenomenological approach to its estimation. Phenomenological models are strictly descriptive in nature. In other words, they can't tell us anything about the underlying mechanism of disease transmission in the population, but their advantage is that they require very little data to parameterize. My group's estimate for R naught was the first on record, but was quickly followed by several others, shown on right through a meta-analytical study we conducted in February 2020. Though there was variability across estimates, as is obvious from this figure, the mean was consistent across studies, indicating that the R naught associated with SARS-CoV-2 in Hubei, China in the earliest days of the pandemic was likely in the ballpark of 2 to 3. As the epidemic in Hubei quickly turned into a global pandemic, however, my group started to wonder just how different R naught was across locations and how the various non-pharmaceutical interventions that were implemented around the world would impact transmission in the months ahead, especially as new waves of transmission began in response to calendar events like this past holiday season. Given the complexity of the new questions we were interested in, we decided to take a more complex machine learning approach in the form of agent-based modeling to address them. However, ABMs haven't been the only machine learning application my group has explored within the context of the pandemic to date. Prompted by how quickly the basic reproduction number studies shown on the right of this slide were produced back in early 2020, we also became increasingly curious about the speed at which the scientific community was working to fill existing knowledge gaps to better understand the novel coronavirus. We were especially interested in understanding whether certain fields within the broad umbrella of something that I like to call outbreak science or outbreak research were experiencing more rapid information generation than others. So, to answer this question, we turned to natural language processing, or NLP techniques like principal component analysis and latent data allocation to sift through the thousands, and I truly mean thousands, of new studies that have been produced in response to the pandemic. During this bibliometric analysis, though, my group became increasingly curious about how the gaps in knowledge might also lend themselves to the proliferation of misinformation. Because of the political context of the pandemic in the United States, particularly during the Trump administration, Americans have been particularly vulnerable to misinformation pertaining to the pandemic. Thus, with an eye towards developing better monitoring and mitigation strategies for public health misinformation, my group tapped into novel digital data streams like news media data and search query data to investigate misinformation events here in the US. These three broad research areas, ABMs for epidemic dynamics, PCA and LDA for bibliometrics, and news media and search query data for event-specific surveillance of misinformation events, comprise a few of my group's core scientific interests. And today, I'll be covering 3 studies from the COVID-19 era that offer a taste of the work that we're doing within each of them. So without further ado, let's get started with ABMs for epidemic dynamics. The paper I'll be presenting in this research area is cited at the bottom of the slide for those who are interested in reading it. In this paper, there were 3 big picture research questions that we aimed to answer through our agent-based modeling approach across 3 different populations of interest: Hubei, China, where the pandemic began; Lombardy, Italy, one of the hardest hit locations worldwide in the earliest days of the pandemic; and New York City, one of the hardest hit locations in the US back in 2020. Recalling the context dependence of the transmissibility potential associated with the given pathogen, the first question we wanted to answer was, how does R naught vary across locations? Second, recalling issues with documentations of infections that I mentioned at the forefront of this presentation, how does documentation rate vary across locations? And finally, recalling the number of non-pharmaceutical intervention policies that have been deployed around the world since the beginning of the pandemic, interventions that we're still employing in some regions today due to lack of vaccine access and emergence of variants, what is the impact of varying policy recommendations during a new wave of infections across locations? Our choice of an agent-based modeling or ABM approach to answer these big picture questions is perhaps best explained by digging into what an ABM actually is. ABMs in our use case can be best described as mechanistic infectious disease models that simulate individual people or agents who are assigned predefined characteristics based off of existing data on the population or populations of interest, including things like region-specific contact matrices, household structure, age, sex, and comorbidities, among other things. The description cards shown on this slide describe some of the attributes that are defined for a given agent in our model to address the aforementioned variability that we expect across populations for 3 questions of interest. By defining each agent's characteristics based off of population-specific distributional data, we're able to simulate epidemics within a synthetic version of each population, including how interventions may curb disease transmission in said populations. Clearly, ABMs require a lot more data and computing power to parameterize than simpler models, which is part of why we didn't use this kind of approach right at the beginning of the pandemic. Unlike simpler models, though, they can lend important insights into more complex questions like the ones we're interested in answering for this study. To simulate epidemics in each of our populations of interest, we fed the agents for any given population through a susceptible, exposed, infectious, and removed, or SEIR style model, as is shown here. We used contact matrix data and household structure data specific to each population of interest to inform transitions from the susceptible state to the exposed state, and we further partitioned our infectious state into 4 categories: pre-symptomatic, mild, severe, and critical, each of which were also assigned a probability of transitioning to one of the 2 removed state categories, recovered or deceased. Folks on the line are probably familiar with SEIR models of the traditional compartmental variety, which are typically defined by ordinary differential equations, or ODEs, and treat each member of a given compartment as identical to one another. In these simpler models, all members of the susceptible compartment have the same probability of transitioning into the exposed compartment. All exposed members have the same probability of transitioning to the infectious compartment, so on and so forth. These kinds of simple models are popular because they are much less computationally intensive and complex than ABMs. However, they aren't optimized for taking into consideration intervention policies and their impact on transmission, an issue that is particularly important for our 3 populations of interest, given shelter-in-place orders that took place at each location early in the pandemic. In other words, they can't capture the individual level variability that make agents different from each other. Something that's important to bear in mind, given the fact that intervention uptake occurs at the individual level. Just because an intervention policy is enacted in a given population doesn't mean that every group within the population will comply with it. This was notably critical to our research interests because we wanted to specifically investigate the impact of age group specific sheltering in place and physical distancing on transmission dynamics during future waves of spread in Hubei, Lombardy, and New York City. To parameterize the SCIR model shown on the previous slide, we used a combination of empirical estimates and free parameters, as is shown in this table. Notably, the probability of infecting an out-of-household contact is a free parameter that plays an important role in estimating R0 across locations. Distributions for free parameters, like the one we're estimated here using a Bayesian inference framework by fitting depth trajectories for each population of interest against ground truth depth trajectory data. Depth trajectories were used as opposed to case counts over time because deaths have generally been a more reliable indicator of outbreak progress across locations throughout the pandemic. Though deaths obviously lag behind infections, they're more likely to be documented than non-fatal cases of COVID-19. That said, we did use ground truth data on case counts to estimate the documentation rate across locations by dividing reported case counts by the number of infections as ascertained from our ABM for each location. Our ABM fits against the ground truth daily death data for each population, shown in small black dots in the 3 panels in this figure, were quite good across locations. The green line shows the median trajectory from our ABMs, and the light blue individual samples from the thousands of simulated epidemics that we ran for each location. The red dotted line indicates when ABMs for each location initiated a reduction in contact across the population in response to non-pharmaceutical intervention policies such as shelter-in-place orders. These policies were implemented at different times across locations, as is clear from the x-axis for the 3 panels. The factor by which contact was reduced following shelter-in-place orders varied by locations. While information on this parameter was determined empirically from a survey for Hubei, it was treated as a free parameter for Lombardy and New York. We did, however, compare our estimates for contact reduction in New York against mobility data from Unicast and found that our estimates were similar. To further validate our fits against death trajectory ground truth data, we conducted several sensitivity analyses under varying assumptions of under-documentation for deaths, as well as out-of-sample validation, as is detailed in the supplementary materials of this particular paper. Once we felt assured of our fits against the ground truth death trajectory data, we were able to estimate R0 for each location, as well as the documentation rate. Substantial differences in R0 are visible across locations in the early days of the pandemic, as shown in the top left panel, with medians of 2.23 for Hubei, 2.95 in Lombardy, and 3.2 in New York City, thus demonstrating important population level variability. R0 values, as estimated through a sensitivity analysis in which deaths were assumed to be underreported in Lombardy and New York City, which, unlike Hubei, were still developing outbreaks at the time of analysis, is shown in the bottom left panel. And notably, the relative order of magnitudes across locations remains unchanged. Substantial differences in documentation rate are also visible, as shown in the top right panel, which with consistently low documentation rates across locations, rates that are further reduced when conducting a sensitivity analysis in which deaths in Lombardy and New York City are once again assumed to be underreported, as is shown in the bottom right panel. Despite low documentation rates across locations, however, all 3 populations remained vulnerable to a new wave of infections as of this time last year, which is when this paper was in preparation for publication. More specifically, our models suggest that only 1.3% of Hubei, 13.8% of Lombardy, and 22% of New York City were infected by the end of our modeling periods for each location, the latter of which has since been confirmed empirically via serological survey. Given the susceptibility of all 3 locations to further transmission of SARS-CoV-2 last summer, we investigated the possible impact of various shelter-in-place strategies to curb transmission in the event of a new wave prior to the widespread availability of vaccines, with a focus on age group-specific interventions. Here, each column shows a different level of physical distancing by the population of interest, where contacts between all age groups are reduced to the given percentage of their starting value. The x-axis within each plot shows the result when the given fraction of a single age group shelters in place, in addition to physical distancing by the rest of the population. The result of this combination of sheltering and distancing is represented by a bar, where the color of the bar indicates the age group which engaged in sheltering, as shown in the key. The height of the bar yields the total fraction of the population infected or the number of deaths in the population in that scenario. Each row gives the results for a single location, where the first 2 plots show the fraction of the population which is newly infected in the new wave, and the next 2 plots show the number of new deaths which occur. We found that sheltering in place by a single age group had the potential to produce a protective effect against transmission for the entire population, but which age group had the greatest impact varied across our 3 locations, thus highlighting the importance of capturing heterogeneity both within and across populations. Furthermore, the impact of sheltering in place was itself impacted by the level of physical distancing undertaken by the rest of the non-sheltered population. Here, 50% contact and 75% contact rates, which represent 50% and 25% reductions in the number of contacts, are shown, with starkly different outcomes with respect to infections, shown left, and deaths, shown right, in our three populations of interest. This work suggests that partial population sheltering rather than universal lockdowns, combined with physical distancing by the remainder of the population, may be a creative solution for reducing COVID-19 transmission in settings where vaccination has yet to reach the population at large, or in the future when variants of concern may be capable of vaccine escape, but that recommendations regarding who should shelter must be population specific. Given the impact of physical distancing as a supplement to partial population sheltering, we also provided a list of policy recommendations to improve the feasibility of physical distancing in our paper, recommendations that are still relevant today in under-vaccinated regions and areas facing new outbreaks of more transmissible variants. These include, but are not limited to, the implementation of shift work, staggering of work shifts, and cohorting of workers in workplaces. One-way aisles and age group-based shopping hours for essential businesses such as grocery stores, pharmacies, and so on. Staggered admission for non-essential businesses such as gyms and restaurants, and remote worship services for religious congregations. Further recommendations, including the feasibility of sheltering in place within the work from home paradigm and so on, are elaborated upon in the supplementary materials as well for those who might be interested in learning more. This all said, though our agent-based modeling structure has been peer-reviewed and published, we're still working on improving it through related and ongoing work that builds off of the structure I presented today and applies it to other locations of interest. Internationally, we've thus far expanded our model to India, and domestically, we've started work on developing state-level models that incorporate occupation into the agent characteristics as defined for each population of interest. Both here and abroad, we're also working on modeling more equitable vaccination deployment strategies that exploit transmission heterogeneity. The fact that not all individuals are as likely to contract or spread SARS-CoV-2 as others, to inform risk-based immunization, particularly in regions with a limited supply of doses, as well as within the context of future booster shots that may eventually be needed in the face of emerging variants. Without question, this is an active area of research where we expect to continue improving our published work to better inform the way that we make policy decisions moving forward, and we're excited to see where our ongoing work takes us. Now, let's dive into another research area of interest at the intersection of machine learning and the COVID-19 pandemic: natural language processing for bibliometric analysis. As I mentioned at the beginning of the presentation, my group became particularly interested in understanding the evolution of research pertaining to COVID-19 due to how rapidly studies, including our own, have been produced in response to the myriad knowledge gaps that exist due to the novelty of the pandemic. More specifically, we were interested in answering three specific questions. First, given that COVID-19 has caused a multi-million casualty pandemic and related coronavirus diseases like Middle East Respiratory Syndrome, or MERS, and Severe Acute Respiratory Syndrome, SARS, thus far haven't, does the research that's been produced for COVID-19, especially given the speed at which it's been produced, differ in some way from research that's been produced for MERS and SARS? Second, what fields or topics were overrepresented and underrepresented in the COVID-19 literature that was published in the first few months of the pandemic, either as peer-reviewed papers or as preprints prior to the peer review process? And finally, how is the focus of COVID-19 research, namely the topics and fields investigated, changing over the course of the pandemic? There are a lot of cool results I want to share today that start to address these three questions. So I'll only offer a very cursory look at the methodology we used in this particular study before jumping into our findings. The paper is cited at the bottom of the slide, though, for those who would like to dig into the more technical aspects. To start addressing your questions of interest, we turn to the COVID-19 Open Research Dataset, or COVID-19. a growing resource of scientific papers on COVID-19 and related historical coronavirus research. As of May 28th, 2020, which is at this point over a year ago, COVID-19 was already comprised of over 130,000 research papers. To save computing time, we decided to restrict our analysis to abstracts. And of the research papers in the dataset, about 100,000 had abstracts available for analysis. Among these, about 35,000 mentioned coronaviruses explicitly in the abstract, indicating that the paper itself was about coronaviruses rather than simply referencing coronaviruses in passing, or say in the citation list. Over half of these coronavirus abstracts were about COVID-19. The remaining fraction referenced other coronavirus diseases, like MERS and SARS instead. After the filtering process shown on the previous slide, we used the resulting 18,412 COVID-19 abstracts and the 16,869 abstracts mentioning SARS and MERS as a source text, or a corpus of documents for our analysis. Natural language processing was implemented on these 35,000 abstracts in two different ways. First, we conducted dimensionality reduction using principal component analysis, abbreviated as PCA. PCA identifies patterns of variance or principal components, abbreviated as PCs, that differentiate documents from one another, highlighting key trends in the data. For example, in a simple corpus with two mutually exclusive topics, such as machine learning and public health, the terms machine and learning would be correlated with one another. PCA would recognize these terms as an important source of variation, providing a way to differentiate documents about either topic, machine learning versus public health, by the frequency of these terms. Following PCA, we conducted topic modeling using latent Dirichlet allocation, abbreviated as LDA as well. LDA is an unsupervised probabilistic algorithm that extracts hidden topics from large volumes of text. Once trained to discover words that separate documents into a given number of topics, LDA can estimate the mixture of topics associated with each document, or in our case, scientific abstract. These mixtures suggest the dominant topic for a document that is then used to assign a document to an overarching topic category. For example, LDA may separate documents into two topics, one on machine learning and another on public health. And if a particular document's mixture is 60% machine learning and 40% public health, it would assign the document to a machine learning topic category. Findings from our dimensionality reduction via PCA suggested that the second principal component does a nicer job of differentiating literature about COVID-19 from literature about MERS and SARS, shown on the right. It's worth noting that earlier PCs usually capture more variance than later PCs, but in this case, we see that PC2 captures more variance because of certain aspects of pre-computation processing that we didn't conduct for the purposes of computational feasibility, as is detailed at length in the supplemental information that accompanies the study. To better understand what kinds of terms were driving the divergence we saw in PC2 between COVID-19 abstracts and abstracts pertaining to MERS and SARS, we examined the component values of the top 50 words on PC2. Here, we see that on the left, lemmatized terms include words like patient, case, and hospit, which represents words such as hospital and hospitalization, among others. Meanwhile, on the right, lemmatized terms include words like virus, cell, and protein. Notably in PCA, words with component values of the greatest magnitude on each PC most strongly drive the pattern that each individual PC recognizes. Here, this means that PC2 detects a pattern such that when protein appears in the text, patient appears less often. To learn more from the lemmatized key terms on the previous slide, we sorted them by their PC2 component values and then examined the percentage of COVID-19 abstracts and the percentage of MERS and SARS abstracts that mention each word. Here, COVID-19 abstracts are shown in orange, and non-COVID-19 or MERS and SARS abstracts are shown in blue. It's immediately obvious that COVID-19 abstracts are overrepresented on the left-hand side, the side that includes words like patient and case, and underrepresented on the right-hand side, the side that includes words like protein and cell. Given that terms like protein and cell are more likely to appear in lab-based research, and terms like patient and case are more likely to appear in clinical modeling and field-based research, this suggests that lab-based research regarding COVID-19 may have been underrepresented in the first several months of the pandemic when compared against MERS and SARS. Digging deeper still, topic modeling via LDA also suggests stark differences in the topics covered by COVID-19 or SARS-CoV-2 related abstracts when compared against abstracts pertaining to other coronaviruses. 30 topics emerged, as shown above, and 5 of these, topic 14, outbreaks impact on healthcare services, topic 15, testing for coronaviruses, topic 17, epidemic cases and modeling, topic 21, clinical care and therapeutics, and topic 25, lessons learned for epidemic preparedness, accounted for 58% of all COVID-19 abstracts and just 17% of MERS and SARS abstracts. In other words, COVID-19 abstracts were disproportionately concentrated in these 5 topics related to non-COVID-19 abstracts. We were also able to group these 30 topics that emerged into a few topic families, namely 1, clinical issues, testing and diagnostics, 2, societies and outbreaks, responses to mitigate them and their impact on society, 3, basic microbiological study, 4, general outbreak reporting, and 5, modeling disease transmission. Temporal analyses of these topic families over time indicates that publications between January 2020 and through the end of May 2020 were dominated by studies involving outbreak response in patients and healthcare services, with no sign of slowing down. Publications regarding viral mechanisms and biomolecular processes related to SARS-CoV-2 grew at a slower pace during this time period, and these results remained consistent when we reran our bibliometric analysis through data that went through July 2020 as well, so almost exactly a year ago. This suggests that as of last summer, COVID-19 research was dominated by clinical modeling and field-based investigation, whereas lab-based studies lagged behind. With the help of colleagues and collaborators who conduct laboratory-based science, we were able to determine that the most likely cause for the lag in lab-based research for COVID-19 has been due to massive federal funding cuts that basic science labs have experienced over the last several years. In other words, these lags are not at all the fault of basic scientists. Instead, they represent insufficient resources to do the work that is essential to pandemic response, including development of improved diagnostics and condition-specific therapeutics. As a result, one of our primary policy recommendations coming out of the study is the need for federal funding mechanisms to prioritize lab-based research, both in the short term for emergent crises like the ongoing pandemic, and through longer-term mechanisms to enable a position of preparedness for future emergent crises. Since publication of this study, numerous lab scientists have reached out to my group and thanked us for providing quantitative evidence for their anecdotal experiences, which they hope will help motivate reevaluation and redistribution of federal funding to lab-based research activities. Indeed, we foresee that our bibliometric analysis pipeline can be utilized by federal funding agencies over time to identify possible special interest areas too, including persistently understudied topics not only in basic science, but within clinical, modeling, and field-based science as well. As a result, we are keeping our pipeline up to date and will continue running it on the COVID-19 data set in the future to see what else we can learn about the information we're creating in response to the pandemic. We also plan to expand our bibliometric analysis pipeline to include clinical trial abstracts to unearth potentially understudied outcomes associated with COVID-19, such as the long-term effects of COVID-19 or long COVID, which to date remain poorly understood and have often been overshadowed by effects associated with therapeutics that are currently undergoing trials. Beyond bibliometric analyses, we would also like to further examine COVID-19 and other scientific literature databases using network science techniques to investigate the COVIDization of public health research. COVIDization of research may be defined as a phenomenon in which researchers who have not previously studied pandemics, epidemics, or coronaviruses are now pivoting towards COVID-19 research for a whole host of reasons, varying from the availability of emergency funding to things that are more malicious, like opportunism. However, given the importance of domain expertise and experience when conducting research about COVID-19, the COVIDization of public health research has the potential to result in information generation that maliciously or otherwise doubles as misinformation propagation, as is showcased by several high-profile retractions that have taken place since the beginning of the pandemic. Of course, poorly executed scientific studies aren't the only source of misinformation that plagues the pandemic, which leads us to the third and final study that I'll be covering today. In the broad research area of utilizing novel digital data sources for event-specific surveillance, this paper focuses on the public health impacts associated with the misinformation event that I'm sure many of us are all too familiar with: President Trump's April 2020 remarks regarding the off-label use of disinfectants or bleach as a COVID-19 therapeutic. For those who may need a refresher, however, let me read President Trump's remarks for you now, verbatim. Quote, "I see the disinfectant where it knocks it out in a minute, 1 minute. And is there a way we can do something like that by injection inside or almost a cleaning? Because you see, it gets in the lungs and it does a tremendous number on the lungs. So it would be interesting to check that, so that you're going to have to use medical doctors with, but it sounds interesting to me." Following this statement on April 23rd, 2020, my research group was interested in answering 3 specific questions. First, how did President Trump's remarks impact the behavior of the general public with respect to interest in purchasing, injecting, and drinking disinfectants? Second, how long did the impact of his remarks last? And finally, was this misinformation event associated with population level increases and adverse health effects, such as poisonings? Like the bibliometric study, there were a wealth of fascinating findings that we discovered through this investigation. So I'll keep my discussion of the methods brief and focus primarily on the results. However, please feel free to read the paper cited at the bottom of the slide and the technical appendix, which provides a wealth of information for reproducibility if you'd like to learn more. To start addressing these questions, we first turned to internet search query data from Google to get a handle on what kinds of disinfectants the general public was searching for, both before and after April 23rd, 2020. Google search query data are fractional and relative. When interest in a term reaches a peak for a given time period, this means that the term was more popular at that time period, and whichever term in a series of terms was most popular among them peaks at 100. Here, we can see that Lysol was the most popular term among the 9 disinfectants considered, and that it peaked around April 23rd, 2020. Other popular disinfectants included bleach and Clorox, with the remainder peeling away from the pack. With our initial search query analysis under our belts, we curated a set of search terms to represent 4 topics of interest: purchasing, drinking, and injecting disinfectants, as well as poison control, a proxy indicator for possible poisonings following the off-label consumption of disinfectants. As is clear from the search terms considered, we chose to focus on the 3 most popular disinfectants that appeared to peak around April 23rd, 2020: Lysol, bleach, and Clorox. We pulled data from Google search trends for these search terms, but we also pulled news media data pertaining to these topics as well, so that we could implement a debiasing procedure to separate interest in President Trump's remarks due to media-generated interest from true interest in the off-label use of disinfectants. The autoregressive machine learning procedure designed to debias the search query data of media-generated interest is detailed at length in the supplementary materials that accompany this paper for those who may be interested. Prior to debiasing the search query data of media-generated interest, though, we plotted our raw search query data, and the results, which are shown here, were pretty stunning. The action-oriented searches that reflected off-label use of disinfectants, namely drinking and injecting disinfectants, peaked immediately following Trump's April 23rd remarks. Then, rather damningly, interest in poison control peaked the very next day, results that are unfortunately consistent with poisonings reported subsequently, noted by the American Association of Poison Control Centers. Moreover, interest in these topics remained increased for an entire week following the misinformation event, suggesting that while most of the associated impact on population level behaviors occurred soon after the event itself, the impact didn't dissipate immediately. Though debiasing to minimize the news media generated interest in these topics marginally blunted results across the topics of interest, as is shown in these 4 charts, the overarching trends described in the previous slide remain the same. We can't infer causation from this work alone, but the fact that internet search query interest in poison control centers peaked after queries regarding off-label use of disinfectants suggests that search query data could be used as an early warning surveillance system of sorts for poison control centers in the future, a need that may be increasingly dire as false cures and prophylactics for COVID-19 continue to be propagated by those in positions of power. Which brings us to the policy recommendations that my research group made following this study. First and foremost, science communication at multiple scales will be necessary to combat sources of misinformation, including elected officials, even in the aftermath of the Trump administration, at least here in the United States. Second, a surveillance system for public health misinformation should be established using novel digital data sources that allow us to monitor population level behaviors following misinformation events. But by designing such a system specifically for healthcare and safety service providers, decision-making tools like the early warning system for poison control centers that I just mentioned can be effectively brought to fruition. With an eye towards establishing a surveillance system for public health misinformation, my research group is currently pursuing a geographic analysis to accompany the temporal analysis presented today to uncover spatial heterogeneities and population level behaviors, and thus vulnerability following misinformation events, as well as sociodemographic risk factors that accompany such vulnerability to enable hotspot prediction of adverse health effects in response to misinformation events. We're also working on incorporating additional data streams, such as Twitter, to glean deeper insights into behavioral impacts following other misinformation events, including those that pertain to vaccines, which is particularly important right now, given refusal and hesitancy here in the United States and our efforts to increase uptake. And on that note, thank you all so much for listening. I know I've covered a lot of material today, but I hope that the 3 studies covered have effectively showcased a sampling of the advances that machine learning can achieve within the context of the ongoing pandemic and public health more broadly. Now, with all that said, I'd be more than happy to answer a few questions over Zoom, and please don't hesitate to reach out over Twitter or email as well. Thank you all so much.
Thank you. Thank you very much. Fantastic presentation with lots of insights. And I think, yeah, this fits very, very well the whole day of lectures. We had this morning Marcel Salatet talking about digital epidemiology, and then also a hands-on session on actually how to, for instance, download Google Trends query.
Oh, fantastic.
With Python. So this is really, so we can see in practice why this is important, right? You just shown to us. So I think there was a question during your presentation in the chat. So you probably also can see the chat. So I think there was a slightly technical question about on the first, probably the first, no, the second paper you presented about why there was a need to do both PCA and topic modeling.
Yeah, that's a great question. So there are a lot of varying opinions about how many techniques should be employed when you are parsing a given corpus of text to understand the types of topics that are being discussed in that text. So in some ways, this is a technical question, but it's also a philosophical one. So one could make the argument that you could have probably just done PCA and drawn the same conclusion that we got to. The reason that we did topic modeling in addition is because we were interested in understanding not just the terms that were very strongly correlated with, say, COVID-19 abstracts versus SARS and MERS abstracts, but also in understanding the broader overarching topic families that were differentiating the two. And so I think that one of the things that was really obvious from PCA is that, you know, for us, it's probably very clear that if you're talking about proteins and cells, this is probably a lab-based paper versus if you're talking about cases and hospitalizations, it might be a more clinical paper. But I think that one of the things that we especially wanted to do with this paper is we wanted to create a pipeline that was more generalizable, that would account for differences and more emergent topics as the pandemic proceeded. So one of the things that we're doing right now, and as I mentioned, this bibliometric analysis pipeline was run last summer, so it's been almost a year. So one of the things that the journal asked us to do actually is they wanted a one-year update. So we're rerunning all of the work that's been done from the past year. And just as kind of a sneak peek, a lot of the results in terms of the thematic representation of COVID-19 papers remains the same. We still see this kind of flag and lab-based studies, but one of the things that's happening is that we're seeing some more nuance in the types of topic families and topics that are emerging because the corpus has grown so much. So in the last year, we've added another several tens of thousands of papers to this corpus. And that's part of why we wanted to include topic modeling as well. I think that for the initial application, we probably didn't need it to draw the conclusions that we drew. But one of the goals here was to create something that was generalizable enough that it could be rerun on this same database over potentially multiple years. And to do that, I think that we wanted to make sure that we had more bases covered, not just in terms of the term differences that we're seeing between COVID-19 and non-COVID-19 related coronavirus abstracts, but also the topic families that emerge. And these are certainly related, but I think that one thing that's really interesting, especially when you work with clinicians, is that you quickly realize that some terms that seem really obvious divisions for us who, you know, largely computer science folks, engineering folks, physicists, things that seem kind of obvious to us as like, oh, these are clearly different from each other, may not be so obvious from a clinical perspective. And one of the things that one of my clinician friends mentioned actually, who's on this paper, is that the idea of protein not appearing with patient is kind of surprising because there are so many conditions that result in protein in urine, and that's something that you would report in a clinical case study, which would appear in our clinical studies categorization. So there are a lot of, I think, philosophical reasons to potentially expand beyond PCA in this sort of situation. But for our sake, it was really very, very practical in that we wanted to make sure that the bibliometric analysis pipeline would still be applicable in a couple of months or years when we expected there to be more diversity in topics, which there have been from the past year. I hope that answers that question.
And I take the chance just to say thanks for pointing to this resource because also it could be interesting for the students since they're now working on a project work during the summer school.
Yeah, and part of the reason I wanted to leave some more time for questions is because I know that projects are part of the class. So if there are questions pertaining to projects or data sources or things like that, that might be helpful for the class. That's something that I'm really happy to talk about because that's honestly that's the best way to learn, right? Doing really hands-on work.
Yeah, and so we today we started like defining the project works, and I mean mainly starting from data sets, right? So the idea was starting from a data set, and then the class can pick a data set and then try to answer a research question with starting from the data set. We actually, it was me and Nicola Pera, we didn't include bibliographic data sets, but actually this is also very relevant. And actually we didn't have in mind the resource that you just shared, but if this is easy to access,
yes.
I mean, this could be also relevant and maybe of interest for
yeah, I think COVID-19 is a very interesting database. There is a bit of a hurdle in terms of cleaning and really making sure that you're getting the abstracts that you want, which is part of the process that I showed today. So you do need to do some filtration But it's been used enough now that there are a lot of open access workbooks in Python, including our own, that can help speed up that cleaning process. So I would really recommend it for folks who are interested in understanding kind of the bibliometric evolution of our understanding of the pandemic. I think it
If
would
I'm
be.
not wrong, I just shared the link. This is a Semantic Scholar COVID-19, right? Is that yeah the right link?
Mhm.
So that's a good starting point. Great. Yeah, thanks so much. So yeah, I just pasted the link into the chat. Then yeah, there is another question regarding Bayesian inference of the parameters with the ABM, I think.
Yes, this is just a matter of ordering. Yeah, the estimates were done first, and then the simulations were conducted. And there's a really, really long technical appendix for this particular paper that describes this process at length. And I think, yeah, that all of the code should be up. And if there's anybody who wants to rerun it, we really, really welcome that. I think that one of the things that I mentioned too in this particular paper is that whenever you build a new modeling framework, one of the things that you want to see happen is you want to see application to other areas. So if that's an area of interest too, I'm happy to kind of discuss this further offline. But in terms of the ordering of events, right, because this is something that is a more complex approach to modeling epidemics, there was an inference period first and then simulation. And one of the other things that happened too is, we simulated a lot of different policies. Not all of them actually made it into the paper, but a big chunk of them are in the appendix. So I would highly recommend looking at those as well. And then the last thing that I'll say about this particular modeling framework is that we have a preprint coming out soon. I hope that kind of shows how this can be used in the vaccination area as well, because this is one thing that I mentioned during the talk. When we first started working on this model, and this is what's so interesting about this pandemic, when we first started working on this model, I don't think any of us could have imagined that we would have so many highly effective vaccines already. And I think that many of us in early 2020 were trying to understand how we could better model non-pharmaceutical interventions. But as it's turned out, these are still really important interventions that we have to keep in mind even in the post-vaccination era. So one of the things that we hopefully have coming out soon is kind of a mix of these two things, trying to show how you can model the interplay between non-pharmaceutical interventions and vaccine deployment, but also how do you leverage things like the heterogeneity that we see across populations to have more equitable vaccine distribution. So with all of that said, the answer to the question is Bayesian inference first, not simultaneous, and then simulations. But the simulations themselves, I covered some of them, but not all of them. And I would really highly recommend for those who are interested in the technicalities to take a deep dive into the technical appendix. We spent a lot of time on it, and I'm particularly, I would say, even more proud of the appendix than I am of the papers. So I hope that at least points you to the right direction.
Thanks. So while maybe other questions come to the chat, I take the chance so also to ask questions that were asked also during previous lectures. For instance, can you comment on what you think about the representativeness of these new data streams in case of epidemiological studies? I mean, yeah, we know, I mean, about these problems that with bias, and just about your view. And actually, I'm particularly eager to know what's your view on vulnerable groups, because I think you have a specific, very and something that I'm very interested also into studying. How much can we learn about vulnerable groups in epidemics and through digital traces?
Yeah, this is such a good question. And I think that this comes up basically every time you discuss digital data for epidemics, it's been a topic that has been widely discussed for a long time. And one of the things that we always say is that as more folks enter the internet grid globally, these data become more representative. So there are some places where the data are pretty representative, others where it's not. I like to give the US as an example because obviously I live here, but one of the things that we talk about a lot here is that, in terms of the percentage of folks who are on the internet and who are using Google, it's pretty high. But the thing is, there are still underrepresented groups even here in the US, and those groups tend to be marginalized. And so what inferences you can or can't make, I think, have to be really strictly constrained to the groups that you're actually getting data about, right? So I think this is probably the number one place where I think we need to be very thoughtful about the data generation process, and then what we can actually say about the populations that we're interested in studying. So an area where this became really, really palpable during the COVID-19 pandemic is mobility data. So I'm sure that everybody on the line is familiar with the various streams of mobility data that have become kind of ubiquitous during the pandemic. Google has released their own stream. We used Unicast here in the US. In my group, there are many, many others. But the thing is, all of these mobility data are based on smartphones. And we know that smartphone penetration around the world is very far from 100%. Even here in the US, we have populations that have pretty low uptake of smartphones. One of those populations is the elderly. And that's particularly difficult because the elderly were the first group to get vaccinated here. The elderly have always been at highest risk of deaths and really, really high morbidity after SARS-CoV-2 infection. So now you have a situation where your most vulnerable group, arguably, is also one of the least likely to be giving you data in terms of where they're going and what they're doing day to day. And I think that this is actually something from a methods point of view, we've had to learn how to adjust for. So one of the things that we've been doing for our vaccination related work is we've been trying to understand how we can use mobility data to understand uptake of non-pharmaceutical interventions. So for example, can we use smartphone data on where people are going to understand whether people are complying with shelter in place orders or whether people are complying with, reduced movement to non-essential businesses or so on and so forth. And so in theory, if these data were representative and everybody had a smartphone and everybody had their GPS location turned on, it would be a really good source for this information. But the reality is it's not representative. So how do you start to infer who is missing, and how might the people who are missing affect the trends that you're seeing in the mobility data? So I think that there's actually an entire area of inference that's now being born out of our understanding of the fact that representativeness in these data sources is not sufficient. And that's in some ways a very good thing. I think it's teaching us to very actively deal with the problem instead of just saying, well, it's a limitation. Like, of course it's a limitation, but there are also ways that we can start to think about, more holistically and systematically, okay, if the elderly are more likely to be missing from mobility data, where do we expect them to be traveling more or traveling less? What sorts of adjustments should we make to the mobility data stream to accommodate our understanding of their lifestyle in that system? So I think it's a really interesting opportunity for social scientists who specialize in marginalized groups, especially, to collaborate with computer scientists to understand how we can integrate our knowledge of these systems and produce adjustments to these data streams. So that's been a really, really interesting, I think, component of all of this. And I didn't dive too deep into our group's use of mobility data, but I think that that's been a really, really common one. I think the other one that I'll mention are contact tracing apps. So I'm sure that everybody on the phone is aware of kind of the myriad contact tracing apps that have appeared around the world. And one of the things that's happened, and part of the reason that they haven't been successful, is that uptake has been really abysmal. And so there are a couple of reasons for this. One of them, again, being that for the most part, they require smartphones. And as I just mentioned, there are plenty of groups that don't own smartphones around the world, including in the US. But there are other reasons too. So privacy is a really, really big issue. And it's something that impacts marginalized communities more. It's something that a student of mine and I have been studying at length during the pandemic. And this idea of being privacy poor. So there are some groups around the world who don't have as many privacy rights as everybody else. Here in the United States, that includes incarcerated people, it includes immigrants and refugees. And so these groups are almost, and I mean, they're predominantly going to be lower socioeconomic status. They're also predominantly going to be Black, Indigenous, and people of color. So then you have these intersecting types of marginalization that you have to consider. So then the question is, okay, if we're requiring people to download contact tracing apps, or more recently, if we're requiring people to download vaccine passports onto their phone, who are we leaving behind? And so my student and I actually wrote an editorial about vaccine passports, particularly discussing this. And, you know, I think that we're all very eager to reopen safely here in the US, at least, where we have, you know, pretty high penetrance of vaccination in a lot of regions, but low penetrance elsewhere. And I think that at the same time, we have to be really thoughtful about how we do this, because this idea of who generates data and why people choose not to generate data, so even folks who have the ability to generate data, why they may choose not to, I think is a really important question in all of this. And so this gets into, I think, a lot of the ethics and policy intersections of all of this. And I think it's very vitally important, but maybe doesn't get as much attention as it should. I remember talking to a class about vaccine passports and discussing this idea that, you know, not everybody has a smartphone, not everybody wants to share these data because they may be fearful of getting deported, or they may be fearful of being fired, and so on and so forth. And it was remarkable to me how few people actually had thought about that. And I think that, you know, it's not necessarily the fault of computer scientists or physicists who aren't necessarily exposed to these sorts of things on a daily basis in their research, but I think that it really drives home the importance of interdisciplinary research during pandemics, because these are things that social scientists and anthropologists, lawyers have been thinking about long and hard for a long time. And this is where integration with those groups becomes really important.
Thank you. Thanks. Yeah, very insightful. There are a couple of other questions. Again, one it's about availability of the code. Mm-hmm. The number of confirmed cases in the model.
Yes. Okay, so all of the code for everything that was presented today is available online on GitHub. It should be linked in each paper. So if you open up the paper, and I will also circulate the slides so it's just easier to find them. If you open up the paper, the GitHub should be linked in each one. And so you should be able to download the code for everything. If you run into any problems, email me or email the first author on any of these papers, and they will get back to you right away. We're very, very excited to have people reusing work that we're doing, because it saves everybody a lot of time. So please feel free to do that. So that's for the first question. The second question is, for the epidemiological models you showed, did you use the number of confirmed cases or any other measure like incidence? Yes, good question. So one of the things that I mentioned kind of in fleeting passing, because I wanted to leave enough time for the Q&A, so I'm glad that this came up now, is this idea that cases have not been a very accurate measure of epidemic dynamics throughout the pandemic anywhere, including in the US, where we arguably have better testing than other places do. And one of the reasons for this is this phenomenon of who we test and who we choose not to test. And this has happened. It's happened to be very variable throughout the pandemic. So we're seeing this right now, even with vaccination, where if you're a vaccinated person and you feel like you have mild COVID symptoms, it is actually still pretty challenging to get a test here. And part of the reason is we're not documenting breakthrough infections of all kinds in the US. We're only monitoring breakthrough hospitalizations. So this is the newest rendition of who we test and who we choose not to test. But throughout the pandemic, there have been lots of variability in terms of who we test, who we choose not to test. And what that means is that our case counts over time are definitely not representative of the epidemic dynamics that are happening in a given place at a given time. And especially if you try to track things over time, you run into a lot of issues, at least here in the US, where when testing procedures will change, suddenly you see this big uptick in cases, but it's not really that the epidemic dynamics have changed at all. It's just that testing practices have changed. The inverse happens too, where testing might become more restrictive, and now suddenly it looks like you don't even have an epidemic when you're looking at cases, but that's not true either. So with all of that said, we actually didn't use cases as our ground truth data. We used depths instead. And so the reason for using depths is that generally, and this is true in most parts of the world, but there are some, I won't say exceptions, it's just that it applies less in some places than others. But in the 3 locations that we looked at, Hubei, Lombardy, and New York City, there was a pretty strong practice of if someone died and they had respiratory symptoms, that they were going to be tested for COVID-19 after a certain point in time. So by the time that, March rolled around, March 2020, we felt pretty confident that that was the case. So that's why we ended up using deaths. Now, obviously, deaths lag behind infections. They also lag behind hospitalizations and cases. So one of the reasons that people may not want to use deaths is because you have to incorporate more time into your modeling period, and that can, create delays of all kinds. But at the same time, we felt a lot more confident in the death data than we did in the cases data. I think that we knew that the cases were severely underdocumented, and we were able to establish this when we looked at the number of infections that our ABMs produced and compared them to the number of cases that were being reported on the ground. And this is something that people have been trying to do lots of different ways, right? Trying to ascertain the documentation rate. I would say most official way to do this is to do serological surveys and to just go around testing everybody to see who has actually been infected with SARS-CoV-2, and then do a comparison from that location to the number of cases that were reported from that place. And that's happening, but these are expensive. So one of the places that this has happened is New York City. And so that study came out a couple of months after our ABM study, and we found pretty comparable results. And so one thing that's really interesting about this is that it suggests that ABMs like this can be used to start estimating our documentation rate and also the total number of infections that have occurred across different locations without needing to deploy really, really expensive serological survey studies that, require not only a lot of money and resources in terms of labor, but are also time intensive. So all of that to say, we used death trajectory data to serve as our ground truth. And the reason that we didn't use case data is because we felt really underconfident in what the documentation rate was like. And one of the things that we wanted to actually estimate was the documentation rate, because that's been of vital importance. One of the reasons this is really important is that it tells us just how many people are getting sick in the population and maybe transmitting to other people, even if they're not showing up in a doctor's office. And this is something that we have a lot of experience with flu. It's a very, very common for flu for people not to seek care, but they're still getting sick and they're still infecting people. And so this is part of what inspired that. And one of the things that we use flu death data for is something very similar to this, where you are making some inferences about what percentage of people you expect to die from the given infection, given a lot of other attributes that they have, their age, their comorbidity status, their sex, so on and so forth. So all of this to say, I think that there isn't any right way to do this. I think that there have been a lot of different approaches to using different types of ground truth data for epidemic modeling, but deaths have been very common. So the death trajectory data, using that as your ground truth, has been very common across lots of different modeling platforms for all the reasons that I've just described.
Thank you. Thanks so much. I think, yeah, we are at the end. I don't know if there are more questions, of course. Feel free to type them. Yeah, it's the end of a long day for the class, of course. But I think, yeah, that was very, very insightful in great series of examples of what does it mean to analyze digital trace data for the COVID-19 pandemic. So I'm really grateful for your presentation, Maya. Very, very helpful, and I'm sure also all the students got a lot of suggestions and hints for possible project work and for their research. So yeah, if there are no more questions, then I can just say thanks so much for for being here with us today. Thank you for your presentation. And well, have a good day. It's still morning
Yes.
on the East Coast. So, and
yeah, thank you for having me. Yeah, and I will send around the slides. I just saw the question in the chat, so I'll send them around.
That's awesome. Yeah, if you want to forward them to me, I will just share them with everyone. We have a shared Google Drive and yeah, that's great.
Okay, sounds good.
Thanks so
much.
much.
All right, fantastic. Thanks, everyone.
Have a good day. Bye. Bye. Okay, great. So we are at the end.